Welcome back to the deep dive. This is where we plunge into those fascinating, sometimes really complex corners of human knowledge. And we try to emerge with, well, clarity and hopefully some of those really illuminating aha moments for you. Today, we're venturing into the intricate landscape of our own biology, specifically the immune system. It's this incredible, just tireless network, isn't it? A guardian designed to protect us from, well, countless threats. But what happens when this guardian, in ways both surprising and incredibly complex, begins to turn against us? That's precisely the profound mystery we're going to try and unpack today. We'll be exploring some of the most intricate mechanisms of immune dysregulation. Now, our mission on this deep dive is a very specific one. It's actually rooted in a critical medical question. We're going to thoroughly explore whether IgG4-related diseases might be, uniquely susceptible to severe inflammatory responses, what we commonly call cytokine storms. We'll be scrutinizing a really compelling hypothesis that suggests our immune system's peacekeepers, the regulatory T cells, or Tregs, can dangerously convert into pathogenic pro-inflammatory TH17 cells, especially when there's a lot of a powerful signaling molecule called interleukin-6, or IL-6, around.
Right, IL-6 is key there.
And crucially, we'll examine the foundational role that immune escape plays in setting the stage for this entire intricate and potentially devastating dance. We've compiled a stack of recent research, expert insights that really piece together this sometimes concerning but honestly endlessly fascinating puzzle. By the time we wrap up this conversation, you're going to walk away with more than just a, surface level understanding. You should gain a much clearer, more nuanced appreciation of these cellular interactions, the powerful molecular messengers driving inflammation, and the incredibly delicate balance that our immune system must constantly maintain.
It's a very fine balance.
A balance that, when lost, can have profound systemic consequences, especially, we think, in the medium and long term. So, let's begin to unravel this complex tapestry. Okay. Let's start by getting acquainted with some of the fundamental cells, the ones orchestrating our immune response. You've probably heard of T cells, right? But what you might not realize is that this isn't just one monolithic group. It's more like a highly specialized orchestra, really, with different sections, different personalities, each with distinct and sometimes contrasting roles.
That's a perfect analogy, actually. T lymphocytes, or T cells, are indeed crucial ringleaders of our adaptive immune function. Adaptive meaning they learn and adapt to specific threats they encounter. They originate from hematopoietic stem cells way back in the bone marrow, but their education, their maturation, that takes place in a specialized organ called the thymus.
The thymus, right.
Yeah. And here they undergo this meticulous, almost rigorous selection process. They're essentially taught to distinguish between self, our body's own healthy components, and non-self, things like bacteria or viruses, foreign invaders. This central tolerance step is absolutely vital for preventing autoimmune attacks later on. Within this diverse T cell population, we prominently have helper T cells. You can identify them by the CD4+ marker on their surface.
CD4+, got it.
And cytotoxic T cells, which carry the CD8+ marker. Now, cytotoxic T cells, the CD8s, they specialize in directly recognizing and killing infected cells, or even cancerous cells. They're the direct fighters. But it's the helper T cells, these CD4+ conductors, that truly play a pivotal role in directing the entire immune response.
So they're not the soldiers, more the generals.
Precisely. They don't fight directly, but they issue the critical commands. They coordinate the efforts of all the other immune cells, making sure the right response is mounted at the right time against the right threat.
So these T cells are like the strategists then. And for any effective strategy, communication is absolutely paramount. How do these cells communicate across this, well, incredibly complex network? I'm guessing it's those chemical messengers we often hear about, the cytokines. Are they the ones allowing them to coordinate such massive effects? What exactly are they, and how do they function as this sort of intricate cellular language?
You're absolutely right. Communication is everything in the immune system, and cytokines are the primary language they use. They are small proteins, almost like molecular tweets or text messages, if you like, released by cells. They function as vital signals to regulate a multitude of biological processes: immunity, obviously, inflammation, and also hematopoiesis.
Hematopoiesis. That's blood cell production.
Exactly. It's the body's internal blood cell factory, constantly churning out all the different components our immune system relies on. Now, to give you a sense of their diversity and specific roles, let's think about macrophages. These are formidable immune cells, the big eaters, basically. They engulf and digest microbes. Phagocytosis? Upon activation, macrophages secrete a whole range of potent pro-inflammatory mediators. For instance, they release TNF tumor necrosis factor alpha and IL-1 interleukin-1. Think of these as the body's rapid-fire alarms.
Okay.
They activate the vascular endothelium, the lining of blood vessels, essentially making the vessel walls sticky and more permeable. This allows other immune cells to quickly exit the bloodstream and get to sites of infection or injury. Often triggers fever, too, as a systemic response.
Ah, so that's part of why we get a fever.
It's part of it, yes. Then there's IL-8. That acts as a chemokine, a chemical beacon. Like a siren calling specific immune cells, mainly neutrophils, are frontline bacterial fighters directly to the inflammatory site. And IL-12 is particularly important for promoting specific types of T-cell differentiation, guiding them towards certain effector functions, like, say, leading a charge against viruses. Each of these cytokines has a distinct yet often interconnected role. It ensures a comprehensive and coordinated immune story unfolds.
That paints a much clearer picture, definitely. The sheer precision of these molecular messages is amazing. Now, our sources introduce a specific type of T cell that seems to be a major player, almost a primary instigator, in many chronic inflammatory and autoimmune conditions, the TH17 cells. What makes them so significant? Why are they drawing so much attention from researchers and clinicians?
TH17 cells are indeed a particularly noteworthy subset of those CD4 plus T helper cells we mentioned, and their significance stems directly from their potent pro-inflammatory capabilities. They are really good at promoting inflammation. Unlike the peacekeepers we'll discuss in a moment, TH17 cells are essentially specialized inflammatory conductors. They ramp things up. Under various inflammatory conditions, these cells are known to produce a whole suite of powerful cytokines. Their namesake, IL-17, is a crucial direct mediator of inflammation. It's a potent signal.
IL-17 from TH17 makes sense.
Exactly. It recruits other immune cells, like those neutrophils we talked about, to the site. It promotes the release of further inflammatory mediators. And in uncontrolled situations, it can drive significant tissue destruction. They also produce IL-22, which plays complex roles, sometimes tissue repair, sometimes antimicrobial defense. But it can also contribute to inflammation by promoting cell proliferation and immune responses in barrier tissues like the skin or gut.
So it's not entirely simple.
Rarely is in immunology. Interestingly, they can even produce interferon gamma, which is a cytokine usually associated with a different T helper subset, TH1. This shows they have considerable plasticity. They can adapt. But the key takeaway here is that when TH17 cells are activated and producing these cytokines, they are essentially signaling for a robust, often aggressive, sometimes destructive inflammatory response. They push the system towards acute and chronic inflammation.
Okay, so when we see TH17 cells active and, flourishing in the body, what kinds of conditions are we usually looking at? Where do their inflammatory effects really make their impact felt most profoundly?
Well, the scientific data strongly associates TH17 cells with numerous human diseases, specifically those characterized by chronic tissue destruction and autoimmunity. Their central pathogenic role is pretty well established now. This includes conditions like psoriasis, where you get that overactive immune response driving excessive skin cell growth, leading to those characteristic plaques.
Right, I know people with psoriasis.
It's quite common. They're also heavily implicated in various forms of arthritis, psoriatic arthritis, which often goes with psoriasis, and ankylosing spondylitis, involving chronic, debilitating inflammation of the joints and spine. In neurological disorders, they play a role in diseases like multiple sclerosis, MS, where the immune system mistakenly attacks the myelin sheath around nerves.
Oh, wow, MS too.
Yes. And critically, for digestive health, they are central to inflammatory bowel diseases, IBD like Crohn's disease and ulcerative colitis. There, chronic inflammation damages the lining of the gastrointestinal tract. In fact, the evidence for their involvement is so strong that it's directly led to successful therapeutic strategies.
Like actual treatments.
Absolutely. Therapies that specifically block the IL-17 pathway or the IL-23 pathway, which is critical for TH17 development and function, have shown remarkable effectiveness in treating many of these conditions. It really highlights just how central and often detrimental these cells are in perpetuating chronic inflammation and autoimmune damage. They're a prime target for trying to calm things down.
It definitely sounds like without TH17 cells, a lot of these chronic conditions wouldn't be nearly as severe, maybe wouldn't even develop at all in some cases. But the immune system isn't just about attacking, is it? It must have incredibly sophisticated mechanisms. That delicate balance you mentioned. This is where regulatory T cells, or Tregs, come in, acting as the counterbalance to all that inflammatory power. They're the peacekeepers, the brakes on the system, right?
Absolutely. Your analogy of peacekeepers and brakes is perfect. That's exactly what Tregs are. They're another critical subset of T cells, but their function is fundamentally opposite, yet equally essential, to that of the TH17 cells. They are unequivocally recognized for their indispensable role in actively suppressing immune responses and, crucially, maintaining self-tolerance.
Self-tolerance meaning not attacking our own body.
Precisely. Think of them as the immune system's internal quality control. They're constantly monitoring and dampening down any immune cells that might mistakenly target healthy host tissues. Honestly, without effective Tregs, our immune system would quickly turn into a self-destructive force. So this delicate balance between the effector T cells like TH17, which drive inflammation and attack,
The attackers.
and the suppressive Tregs, which actively dampen and prevent it.
Yeah.
That balance is fundamental for maintaining immune homeostasis, a state of stable equilibrium. When this balance gets disturbed, for example, if there's a reduction in the number or the function of total CD4 plus FOXP3 plus Treg cells.
FOXP3 is the marker for Tregs.
Yes, FOXP3 is a key transcription factor and marker for Tregs. So if Treg numbers or function go down, and at the same time you see an increase in TH17 cells, as has been observed in conditions like myocarditis and inflammatory heart condition, well, that points directly to a state of profound immune dysregulation.
The brakes are failing.
Exactly. The brakes are failing and the inflammatory response is unchecked. And that is a hallmark of many autoimmune diseases and really the starting point for understanding the deeper hypothesis we're tackling today.
Okay, we've set the stage. We know the key players, the inflammatory TH17s, the peacekeeping Tregs. Now, let's truly dive into the heart of our deep dive, the central hypothesis driving this whole investigation. It's this idea that regulatory T cells are peacekeepers, can somehow convert into pathogenic TH17 cells, the inflammatory drivers, and that interleukin-6, IL-6, might be the critical catalyst, the master switch, for this really profound transformation. This sounds like a point where the whole system could tip from controlled homeostasis into, well, chaotic self-destructive inflammation.
It is indeed a truly fascinating and crucially important mechanism. And it profoundly underscores the plasticity of the immune system, its ability to change. Our sources clearly and consistently demonstrate that interleukin-6, IL-6, plays a central and remarkably multifaceted role. It's what we call a pleiotropic inflammatory cytokine.
Pleiotropic meaning it does many different things.
Exactly. Many diverse effects. Its influence spans across virtually the entire immune system. So beyond its general involvement in acute and chronic inflammation, IL-6 is instrumental in regulating antibody production and even autoantibody production. This directly influences how our body recognizes and responds to antigens, including potentially our own self-antigens.
Ah, so it's involved in autoimmunity directly there too.
It can be, yes. It's also critical for activating T cells and guiding the differentiation of B cells into those antibody-secreting plasma cells. Furthermore, IL-6 significantly increases the production of acute phase proteins by the liver. These are important markers and mediators of systemic inflammation, contributing to that sick feeling during illness.
Right, makes sense.
Its reach extends even further. It impacts hematopoiesis, as we mentioned, but also angiogenesis.
New blood vessel formation.
Correct. Crucial for tissue repair, but also sometimes exploited by tumors. It affects vascular permeability, how leaky blood vessels are, and even osteoclast differentiation, influencing bone remodeling. Given this extensive and diverse involvement, it's really not surprising that IL-6 is implicated in the pathogenesis of numerous inflammatory diseases. It truly acts as a kind of master conductor, orchestrating a vast symphony of cellular responses.
So IL-6 isn't just involved in inflammation, it's a kind of master regulator. It's deeply intertwined with so many processes. It makes it a pivotal figure in this whole immune narrative. But how exactly does it facilitate this Treg to TH17 conversion? Or maybe it's more accurate to say the differentiation of naive T cells into TH17. What's the precise molecular switch at play here that causes this fundamental shift in identity?
Well, the evidence from your sources is quite direct on this. It provides a very clear molecular pathway for this differentiation process. IL-6 specifically induces the differentiation of TH17 cells from naive CD4 plus T cells.
Naive meaning they haven't specialized yet.
Exactly. Uncommitted T cells that haven't yet decided which helper subset they'll become. This process occurs through a well-defined signaling cascade inside the cell. When IL-6 binds to its receptor on the T cell surface, it activates a crucial intracellular signaling pathway involving STAT3.
STAT3.
Stands for signal transducer and activator of transcription 3. Once STAT3 is activated, it travels to the nucleus, the cell's control center, and there it induces the expression of ROR.
ROR gamut. That's the key.
That is the key transcription factor,
Yeah.
absolutely essential for TH17 development. Think of transcription factors as master conductors of a cell's internal orchestra. They don't
Well,
play instruments themselves, but they tell specific genes when and how loudly to play. They essentially dictate the cell's identity and function.
got it.
Orrot is the one that tells a T cell, right? You are going to become a TH17 cell. So in essence, IL-6 provides that initial inflammatory signal that triggers STAT3, which then switches on the master gene ROR, irreversibly committing the T cell to a TH17 fate. This detailed mechanism is really central to the hypothesis we're examining today. It showcases IL-6's direct and potent role in shaping T cell identity.
That's incredibly specific. It really illustrates a powerful molecular switch at work. But as you've emphasized, the immune system is rarely a simple one-way street, is it? It's dynamic, always adjusting. Are there any nuances to this conversion or other factors that might influence it, maybe pulling the T cell in a different direction or modulating the strength of that IL-6 signal?
Yes, you've hit on a crucial point there. The immune system operates with this exquisite dynamic complexity. It's constantly integrating multiple signals. While IL-6 strongly promotes TH17 differentiation, other cytokines in the local microenvironment can significantly influence the outcome. It shows the fine-tuning capabilities of the system. For instance, high levels of another cytokine, TGF-1 transforming growth, factor beta-1, have been observed to inhibit IL-6-dependent TH17 cell differentiation.
Oh, interesting. So TGF-beta works against IL-6 here.
In this context, yes. And this is a critical nuance because TGF-beta is also known for its crucial role in promoting the development and maintenance of regulatory T cells. The Tregs are peacekeepers.
Ah, so TGF-beta pushes towards Tregs while IL-6 pushes towards TH17.
Broadly speaking, yes. So in an environment rich in TGF-1, the scale might tip towards tolerance and regulation, even if there's some IL-6 present. This indicates a complex and ongoing interplay between these cytokines. They're controlling the delicate balance between TH17 and Treg cell populations. The system is constantly adjusting its equilibrium based on the prevailing cytokine environment. The ultimate fate of a naive T cell isn't just dependent on the simple presence of IL-6, but also on the relative concentrations and intricate interactions with other signaling molecules.
It's a real cellular tug-of-war for the T cell's identity, then.
That's a great way to put it, a constant tug-of-war.
That tug-of-war analogy really drives home the dynamic nature of these cellular decisions. Are there experimental observations, maybe from animal models or in vitro studies, that provide strong, direct confirmation of IL-6's crucial role here? Something demonstrating its absolute necessity for this differentiation.
Indeed, there are compelling experimental observations that provide rigorous mechanistic validation. Really elegant studies. Researchers have used naive CD4 plus T cells isolated from mice specifically engineered to lack the IL-6 receptor alpha chain. These are called IL-6 mice.
So they can't see the IL-6 signal.
Exactly. When these genetically modified cells were exposed to direct IL-6 activation in a lab setting, they were shown to be completely unresponsive in terms of TH17 differentiation. They simply failed to develop into the TH17 phenotype, no matter how much IL-6 was present. This clearly showed the receptor is essential.
Okay, that makes sense.
But here's the elegant part of the experiment. This functional deficit was remarkably restored when they introduced a recombinant IL-6 fusion protein, sometimes called hyper-DS SIL6R or HDS.
A fusion protein, what does that do?
It effectively bypasses the need for the missing IL-6 receptor on the cell surface. It provides a soluble receptor complex that can still grab the IL-6 and transmit the signal into the cell even without the cell's own receptor.
Wow, okay, so it proves the signal itself is the key.
Precisely. This experiment provides powerful, direct mechanistic validation. It demonstrates unequivocally that a functional IL-6 receptor and the downstream signaling initiated by IL-6 are absolutely indispensable for driving TH17 differentiation. It confirms IL-6's direct and crucial involvement.
It really sounds like a scientific smoking gun for IL-6's role in this whole process.
It's very strong evidence, yes. Hashtag the positive feedback loop.
What's truly fascinating here, and actually potentially quite alarming, is that this isn't just a single isolated event, is it? It sounds like once these TH17 cells are activated and start producing their signature cytokines, there might be a dangerous, almost self-sustaining feedback loop involved, like an amplifier pushing the system further and further out of control.
You've hit on an absolutely critical point. This transforms a single differentiation event into a potential self-perpetuating cascade of inflammation. It's really key. Our sources highlight a vital and concerning mechanism. Interleukin-17, IL-17, the signature cytokine from TH17 cells, has the capacity to promote autoimmunity, partly by triggering a powerful positive feedback loop through IL-6 induction.
Okay, unpack that. IL-17 makes more IL-6.
Exactly.
Sure.
So as TH17 cells become active and produce IL-17, that very IL-17 doesn't just act on target tissues causing damage. It can, in turn, stimulate other cells in the inflammatory microenvironment cells like macrophages, fibroblasts, even epithelial cells to
Oh.
produce more IL-6.
Ah, I see where this is going.
Right. And as we just discussed in detail, increased IL-6 then further drives the differentiation of more naive CD4 plus T cells into pathogenic TH17 cells. So you get this escalating dangerous cycle. More TH17 leads to more IL-17, which leads to more IL-6, which then leads to even more TH17 differentiation and
It's
activation.
an inflammatory amplifier, a vicious cycle.
Precisely. A self-perpetuating loop of inflammation and autoimmunity. And this profound insight helps explain why immune dysregulation can become so tenacious, so difficult to resolve in chronic conditions. It suggests that maybe just suppressing symptoms isn't enough. We might need to identify and effectively break these feedback loops, perhaps by specifically targeting IL-6 or IL-17 to truly reset the system and regain immune homeostasis.
Otherwise, the system just keeps feeding its own fire.
Keeps feeding its own fire, pushing itself further and further out of balance, especially over the medium and long term.
This talk of escalating, self-perpetuating inflammation, it brings us inevitably to a terrifying phenomenon, one that gained widespread recognition, particularly during the COVID-19 pandemic, the cytokine storm. It sounds like the immune system completely going off the rails, an indiscriminate, overwhelming attack. What exactly defines it? And how does it relate to IL-6 and our TH17 discussion, especially in this context of chronic dysregulation potentially predisposing someone?
A cytokine storm truly represents an uncontrolled, hyperactive, and profoundly dysregulated inflammatory response. Imagine the immune system's alarm bells going off at maximum volume, but with no mechanism to turn them down or restore order. It's just chaos. It's characterized by an overwhelming and amplified increase in the production of various pro-inflammatory cytokines, not just one or two, but a whole flood leading to a rapid cascade of pathological events throughout the entire body.
So it's systemic, not localized.
Absolutely systemic,
Okay.
a body-wide explosion. This includes a rapid worsening and intensification of pathological disorders in the blood coagulation system, leading to hypercoagulability and increased tendency for blood clots to form, as well as that uncontrolled activation of inflammation itself. This extreme state can be triggered by severe infections like COVID-19, certain autoimmune conditions flaring up, or even sometimes by advanced immunotherapies where the immune response becomes excessively aggressive and widespread.
So the body's defense mechanism essentially becomes the primary aggressor.
That's exactly it. The protector turns attacker.
And the consequences are far beyond just feeling very ill, aren't they? It's not just a severe reaction. It can be truly life-threatening, causing immense damage to the person.
Very much so. The uncontrolled activation of inflammation and those coagulation pathways in a cytokine storm can quickly lead to what has been ominously termed a deadly loop. This loop describes a vicious, self-reinforcing cycle. Pathological immunity, the immune response gone awry, leads to pathological inflammation, which in turn leads to pathological thrombosis or widespread blood clotting.
Immunity, inflammation, clotting, a deadly trio.
A deadly trio, yes.
Yeah.
The ultimate outcome of this unchecked process is widespread tissue damage across multiple organs. You get blockage of the microcirculation in these organs, effectively starving them of oxygen and nutrients, and then severe multiple organ dysfunction. This can rapidly progress to organ failure and, tragically, death in many cases. Essentially, the immune system, designed to protect us, enters this uncontrolled overdrive, causing immense collateral damage to the host's own tissues and systems, far beyond any initial threat that might have triggered it.
It's a profound failure of regulation.
Absolutely, where the defense mechanism itself becomes a primary cause of severe systemic pathology. Hashtag the IL-6 connection to cytokine storms.
Okay, so given its role as a master regulator in general inflammation, T-cell differentiation, and especially its role in that dangerous positive feedback loop, where does IL-6 fit into this alarming picture of a rampaging immune response, this full-blown cytokine storm? It sounds like it could be both a trigger and a driver of the whole thing.
IL-6 is unequivocally a key player. It's frequently cited as a predictor, even used sometimes as a diagnostic marker, in the context of cytokine storms. Our sources indicate that detecting a significant augmentation, a sharp rise of IL-6 production in the serum, in the blood, can be very helpful in understanding and predicting the onset and severity of cytokine storms. This was particularly vividly seen in the disease process of COVID-19.
So high IL-6 is a major red flag.
A major red flag, yes. This elevation of systemic IL-6 levels signals a body-wide, uncontrolled inflammatory state that is already well underway or rapidly developing. This really underscores IL-6's crucial role, not just in initiating the differentiation of those pathogenic TH17 cells we discussed earlier, but also in actively driving and sustaining the broader systemic inflammatory response that characterizes a cytokine storm. Its presence in high, rapidly rising concentrations is a potent indicator that the immune system's delicate balance has been severely disrupted. The system is spiraling towards critical dysfunction.
So it's not just a bystander molecule.
Not at all. It's a central orchestrator of the storm's intensity and persistence.
And we've also heard of cytokine release syndrome, CRS, often associated with advanced therapies like CAR-T cell therapy, which is a powerful immunotherapy for cancer. Is that basically a similar event? Perhaps a therapeutically induced cytokine storm, illustrating the same underlying mechanism.
Exactly. You've hit on a perfect parallel there. Cytokine release syndrome, or CRS, is fundamentally a cytokine storm, but often with a more defined, sometimes even predictable, trigger. It's recognized as a significant and potentially life-threatening toxicity in advanced immunotherapies like CAR-T cell therapy.
Where they engineer the patient's own T cells, right?
Correct. A patient's own T cells are genetically engineered to specifically target and kill cancer cells. Now, when these newly introduced, highly activated CAR-T cells rapidly engage their cancer targets, they can release a massive, uncontrolled amount of cytokines, including IL-6, into the systemic circulation. This leads to a systemic inflammatory response that precisely mirrors the clinical and pathological features of other cytokine storms.
So it looks the same clinically.
Very similar. CRS is closely linked to widespread systemic inflammation and can indeed lead to severe clinical outcomes, including organ dysfunction, just like other forms of cytokine storms triggered by infections or autoimmune conditions. This phenomenon powerfully reinforces the concept: excessive, widespread cytokine release, irrespective of the initial trigger. Whether it's a severe infection, an autoimmune flare, or a powerful therapeutic intervention, can precipitate this dangerous systemic inflammatory state. It's the same underlying immune dysregulation, just with different origins. Hashtag TH17's contribution to the storm.
Okay, given how pro-inflammatory TH17 cells are and how their activity can be amplified by IL-6 through that feedback loop, do they actively contribute to these cytokine storms? Are they essentially adding critical fuel to the fire? And what does that imply for how we might potentially treat these severe inflammatory reactions?
Absolutely. TH17 cells are not just passive contributors. They are active and significant participants in fueling these storms. They definitely add fuel to the fire. The existence of a clear TH17 functional phenotype, meaning these pro-inflammatory cells are present, active, and producing their cytokines, has been specifically observed and documented in COVID-19 patients, especially those with severe disease.
So they found them right there in the thick of it.
Exactly. This observation strongly suggests that the host TH17 inflammatory responses directly contribute to the severe lung pathology and acute respiratory distress syndrome, ARDS, often seen in these critical cases. The robust production of IL-17, IL-22, and other inflammatory cytokines directly exacerbates the uncontrolled immune response. It causes further tissue damage and propagates that cytokine cascade.
Making everything worse.
Making everything worse, yes. This understanding is so profound, in fact, that it has directly led to important clinical considerations. There have been suggestions that blocking the IL-17 pathway using existing biological drugs already approved for other autoimmune conditions, like psoriasis or rheumatoid arthritis, could represent a novel and potentially effective strategy to treat patients experiencing severe SARS-CoV-2 infection-induced cytokine storms.
So repurposing existing drugs based on this mechanism.
Exactly. It clearly highlights the critical and pathogenic role of TH17 cells in driving the destructive inflammation characteristic of these life-threatening scenarios. And it offers a targeted approach: calm the storm by removing one of its key accelerants.
Right, now let's try and tie all of this intricate understanding back into the first part of our initial focus today, IgG4-related diseases. First off, what exactly are they, and how do they fit into this complex picture of inflammation, immune dysregulation, and our central hypothesis about potential susceptibility to cytokine storms, particularly in the medium and long term?
Okay, IgG4-related diseases, or IgG4-RD, are a relatively recently recognized group, and quite enigmatic, actually. They're chronic fibroinflammatory conditions. What makes them so distinct is their ability to affect nearly any organ system in the body.
Really? Any organ?
Pretty much. From the pancreas, salivary glands, tear ducts, to the kidneys, aorta, lungs, retroperitoneum, The list goes on. They're often characterized by a distinctive histopathological pattern, what the tissue looks like under a microscope. You see dense infiltration by lymphocytes and plasma cells, especially plasma cells that are producing IgG4 antibodies.
Hence the name IgG4 related.
Precisely. And often, though not always, patients have elevated serum IgG4 levels in their blood. Now, while these are distinct clinical entities, our sources provide a crucial insight into how IgG4 itself, which is one of the 4 subclasses of immunoglobulin G antibodies, might play a broader role in immune pathogenesis. It seems to have some unique structural properties, particularly within its constant region, the tail of the antibody. This is important because it hints at a mechanism through which these diseases, by their very nature, might generate a specific, sustained inflammatory environment that could prime the system for further dysregulation down the line.
That's a fascinating point about IgG4's unique structural properties. Can you elaborate on that role a bit? How does IgG4 actually contribute to immune-mediated disease processes, especially given it's often thought of as being relatively benign, maybe even anti-inflammatory sometimes?
You're right. IgG4 is often considered somewhat unique among antibody subclasses. It sometimes even has anti-inflammatory or blocking properties in certain contexts, like allergy immunotherapy. However, one of our sources highlights a really fascinating example of how it can potentially turn pathogenic. It discusses rheumatoid factor, or RF-like Fc binding reactivity.
Okay, what does that mean, RF and Fc binding?
Right. Rheumatoid factor is an antibody that binds to other antibodies, often seen in rheumatoid arthritis. The Fc region is the constant tail part of an antibody molecule. This research suggests that the gamma-4 constant region of the Ig4 antibody itself can confer this RF-like Fc binding reactivity. It's not happening through the classic antigen binding site at the top of the antibody, but through its tail structure.
So the antibody itself is sticky in a problematic way.
Sort of, yes. This research, specifically done in the context of rheumatoid arthritis, RA, a classic autoimmune disease, suggests that increased production of IgG4 might actively contribute to the pathogenesis of RA by binding to other antibodies, forming immune complexes that then promote inflammation and tissue damage. What this indicates is that IgG4, through these inherent structural properties of its constant region, isn't always benign or anti-inflammatory. It can, in certain contexts, directly participate in immune-mediated disease processes, actively contributing to the very inflammatory milieu we're discussing.
Okay, that's a vital mechanistic detail.
It is, because it helps us bridge the gap. If IgG4 can promote inflammation in RA through this unique Fc binding, it implies a similar pro-inflammatory capacity, a sort of baseline inflammatory state, could be present and maintained in IgG4-RD, setting the stage for subsequent immune dysregulation. Connecting IgG4 to the cytokine storm hypothesis.
Okay, so if IgG4 itself, through its unique structural properties, can contribute to autoimmune pathogenesis and a pro-inflammatory state, like in RA, and we know that TH17 cells and IL-6 are driving factors in cytokine storms and other autoimmune diseases, how does this all logically connect to our core hypothesis for IgG4-related diseases? Are they, by their very nature, potentially more susceptible to cytokine storms, particularly through that Treg to TH17 conversion mechanism you detailed, driven by IL-6, especially looking at the medium and long-term risk? This logical bridge is crucial for our central mission today.
This is precisely where we try to bring together all the threads of our hypothesis, moving from those specific cellular and molecular mechanisms to a broader, potentially clinically relevant implication for people with IgG4-related diseases. Now, to be clear, our sources don't explicitly state that IgG4-related diseases are definitively more susceptible to cytokine storms due to a specific Treg to T17 conversion mechanism happening uniquely within the context of IgG4-RD itself. The direct link isn't fully drawn in the literature we reviewed. However, we can certainly construct a very strong logical and mechanistically sound bridge based on the comprehensive evidence provided about the individual components.
Okay, so we're connecting the dots based on established mechanisms.
Exactly, connecting the dots. The crucial point here is, if IgG4, as demonstrated in the context of RA, possesses the capacity to drive immune pathogenesis and contribute to inflammation, and given that IgG4-RD are, by definition, chronic inflammatory conditions, then it's highly plausible that these diseases inherently create and sustain a milieu of chronic inflammation and immune dysregulation. This isn't just a fleeting inflammatory event. It's likely a persistent, maybe low-grade, but chronic inflammatory state that fundamentally alters the immune microenvironment over the medium and long term.
Okay, so if this baseline chronic inflammatory environment is indeed a hallmark of IG4-related diseases, how does that specifically prime the system for something as severe as a cytokine storm, especially via that IL-6 TH17 axis we've discussed? What are the specific connections we can draw?
Right. So if this IgG4 mediated inflammation, or the general inflammation characteristic of IgG4-RD, contributes to an environment where interleukin-6 levels are persistently, maybe even just moderately, but chronically elevated, which, as we know, is a common feature of many chronic inflammatory conditions, and IL-6 is a potent pro-inflammatory cytokine. Then we have the key ingredients for our hypothesis lining up.
The IL-6 trigger.
The IL-6 trigger.
Yeah.
In the context of the established mechanism we discussed earlier, IL-6 driving TH17 differentiation via STAT3 and ROR, this chronic increase in IL-6 could indeed act as that continuous master switch. It would constantly nudge naive CD4 plus T cells towards becoming pathogenic TH17 cells. This constant differentiation pressure leads to a gradual, but potentially significant expansion of the TH17 cell population over time.
A slow buildup of these inflammatory cells.
Yep. Exactly, a slow buildup. Now, couple this TH17 expansion with that potent positive feedback loop we highlighted, where the IL-17 produced by these TH17 cells induces even more IL-6 production. Well, then you dramatically amplify the existing inflammatory state. Over the medium and long term, this creates a situation where the immune system is fundamentally primed. It's constantly operating with elevated levels of pro-inflammatory cells and cytokines. It's inherently less stable.
Like it's closer to the edge already.
Closer to the edge. This escalating cycle would then significantly increase the susceptibility to a widespread systemic inflammatory response, potentially culminating in a full-blown cytokine storm. This risk would be particularly heightened if other underlying immune escape mechanisms, which we need to delve into next, are also at play, providing a ready pool of autoreactive cells to fuel this dysregulation.
So the critical takeaway here is this convergence of pathways. IgG4 possibly contributing to a chronic pro-inflammatory state that consistently feeds into the IL-6 TH17 axis. This creates fertile ground for severe and potentially devastating immune reactions like cytokine storms, not necessarily as an immediate trigger from the IgG4-RD itself, but as a significant long-term predisposer, increasing vulnerability.
All these intricate pathways really highlight how incredibly sensitive and precise the immune system needs to be just to maintain peace within the body. But you mentioned that at the root of many autoimmune problems, and perhaps underlying this chronic susceptibility we're discussing with IgG4-RD, is this concept of immune escape. It sounds like the immune system fundamentally failing its core duty of self-preservation. What exactly are we talking about here? How does this escape happen?
Well, in the context of autoimmunity, immune escape refers exactly to that. The fundamental failure of the body's mechanisms to maintain self-tolerance. Normally, the immune system has this incredibly sophisticated, multi-layered array of checkpoints and regulatory mechanisms. They're designed to eliminate or suppress any immune cells that show reactivity against its own healthy tissues.
Essential safety checks.
Absolutely vital safety checks to prevent a self-destructive attack. Now, when these checkpoints fail, or are perhaps circumvented for various reasons, autoreactive immune cells, those are cells that should have been silenced or removed, they manage to escape these regulatory controls. Once escaped, these cells are then free to initiate or exacerbate disease by targeting host tissues. They lay the groundwork for chronic autoimmune conditions, or they can amplify existing inflammatory states, like we hypothesize might happen in IgG4-RD.
So it is, as you said, a profound and fundamental weakness. The system's inability to effectively police itself and distinguish friend from foe all the time.
Exactly, like a rogue agent slipping through security and then waiting for an opportunity.
Okay, so if these mechanisms are so sophisticated, where do these crucial checkpoints actually exist within the immune system? Are they at specific locations, or do they act at different stages of immune cell development and life?
They primarily exist at two crucial levels during the lifespan of immune cells. It forms a robust, though clearly fallible, two-tiered defense system against autoimmunity. First, we have central tolerance. This occurs during the very early development stages. For T cells, it's in the thymus. For B cells, it's in the bone marrow. Here, immune cells that show strong reactivity against self-molecules are largely eliminated. The process is called clonal deletion, essentially. They're commanded to undergo programmed cell death, apoptosis.
So the really dangerous ones get removed early.
Mostly, yes. In the case of B cells, there's also a mechanism called receptor editing where they can try and change their receptor to remove the self-reactivity. It's a stringent screening process, like basic training where any recruits likely to turn on their own side are immediately discharged.
But you said largely eliminated. It's not perfect.
It's not perfectly foolproof, no. Some B cells, for example, that are only weakly reactive to self-molecules might be allowed to exit the bone marrow and enter the circulation. These are sometimes referred to as sleeper B cells. They normally remain quiescent, harmless. But under inflammatory conditions, say during an infection, they can become activated and contribute significantly to autoimmune diseases later on.
So that's an initial subtle form of immune escape.
Exactly. Potentially harmful cells slipping through the first line of defense, just waiting for a trigger. Then second, there's peripheral tolerance. This acts as a critical secondary line of defense. It deals with those autoreactive T and B cells that, despite central tolerance, manage to escape into the peripheral circulation and tissues.
So backup systems in the field.
Backup systems in the field, exactly. Here, specialized cells and mechanisms work to suppress these potentially harmful escape cells. Key players in peripheral tolerance include things like tolerogenic dendritic cells, TOL-DCs. These are antigen-presenting cells, but instead of activating T cells to attack, they present antigens in a way that induces tolerance or anergy, a state of unresponsiveness in potentially autoreactive T cells. And very importantly, are regulatory T cells, the Tregs, which we discussed earlier as the peacekeepers.
Right, the brakes again.
The brakes again. They actively suppress effector immune responses through various mechanisms direct cell-to-cell contact, secreting immunosuppressive cytokines like IL-10 or TGF-α, even metabolically depriving effector cells of resources. So even if a self-reactive cell gets out of basic training, there are still mechanisms out in the periphery designed to disarm it or keep it quiet.
Okay, so even with these multi-layered, sophisticated safeguards, central and peripheral tolerance, things can clearly still go wrong. Immune escape happens. What are some of the known triggers or specific mechanisms that actually contribute to this escape and consequently to the development or worsening of autoimmunity? It seems like it's often more than just a random error in the system.
You're right. It's rarely just a random error. There are often identifiable triggers and well-described mechanisms that can overwhelm or bypass these tolerance checkpoints. A key one is molecular mimicry. This is where components of invading pathogens, say specific proteins from the SARS-CoV-2 virus example, share structural similarities. They look molecularly similar to human proteins.
So the immune system gets confused.
Exactly. When the immune system mounts a robust response against the pathogen to clear the infection, it might mistakenly develop antibodies or T cells that cross-react. They recognize and target these structurally similar self-antigens as well as the virus. It's like an immune cell seeing a foreign uniform and a friendly uniform that look almost identical and then shooting at both.
That makes sense. What else?
Another significant mechanism is bystander activation. This happens when an infection or even significant tissue injury causes generalized inflammation and cell damage in a particular area. This inflammatory environment itself, rather than specific antigen recognition, can activate normally quiescent autoreactive T or B cells that happen to be nearby, even if they don't directly recognize the pathogen or the initial cause of damage.
So they get caught up in the crossfire, essentially.
Caught up in the crossfire, activated by the surrounding chaos and alarm signals, and then they start targeting self-antigens released from damaged tissues. These combined actions, mimicry and bystander activation, can initiate a whole series of dysfunctional immune responses, leading to the development or exacerbation of autoimmune diseases. For instance, we've seen increasing reports of various autoimmune and inflammatory diseases following SARS-CoV-2 infections. Things like Guillain-Barré syndrome or multisystem inflammatory syndrome in children, MISC. These clearly underscore these mechanisms at play.
So the infection acts as a powerful trigger, potentially unveiling a latent autoimmunity that was previously kept in check.
Precisely. It tips the balance.
This concept of immune escape sounds absolutely foundational then. It's like the opening chapter to this very complex story of immune dysregulation we've been building. How does it specifically fit into our main hypothesis about IgG4-related diseases, the role of IL-6, the TH17 conversion, and that potential for cytokine storms, especially in creating that susceptibility in the medium and long term? Where does immune escape enter this complex narrative we've constructed?
Immune escape is indeed absolutely foundational. You can think of it as the necessary precursor, the fundamental weakness that really sets the stage for the entire scenario we've been discussing, especially when we consider long-term susceptibility. It provides the initial pool of autoreactive T and B cells. Those are the cells that have, despite all the sophisticated checks, managed to evade both central and peripheral tolerance mechanisms.
The escape.
The escapees, exactly. These escape cells are now primed. They carry the inherent potential for self-harm, but they might remain dormant or maybe just low-level active until an appropriate trigger or environmental change occurs. Now, this is where the entire narrative converges. If such autoreactive CD4 plus T cells are present, having successfully navigated around the body's tolerance checkpoints, and then they encounter a persistently inflammatory environment rich in interleukin-6, an environment that, as we've hypothesized, could be amplified or sustained by an initial IgG4-driven pathogenesis in IgG4-related diseases. And these escaped autoreactive T cells become highly susceptible to differentiating into those pathogenic TH17 cells.
Okay, so immune escape provides the problem cells, and the IL-6 environment activates them down the wrong path.
Precisely. It's a critical convergence point. Immune escape offers the ammunition in the form of these primed, potentially self-reactive cells, while IL-6 provides the trigger and the pro-inflammatory environment for their conversion into highly inflammatory, damaging TH17 cells. These newly differentiated TH17 cells then dramatically escalate the inflammatory response. They're capable of fueling that dangerous positive feedback loop with IL-17 and IL-6 that we talked about. This chronic self-amplifying cycle of inflammation, driven by these escaped, now pathogenic TH17 cells, fundamentally increases the body's baseline inflammatory load and likely reduces its overall regulatory capacity over time.
It erodes the system's stability.
It erodes stability, exactly. Over the medium and long term, this makes an individual, perhaps someone with IgG4-RD experiencing this underlying process, significantly more susceptible to experiencing a full-blown cytokine storm if they encounter an additional strong challenge, like a severe infection. In essence, immune escape isn't just a contributing factor. It's what fundamentally enables the existence of self-targeting immune cells in the first place. And the IL-6 TH17 axis then dramatically activates and escalates them, transforming a potential weakness into a severe systemic threat, particularly over time. #check outro.
Wow. We've truly explored a profound interconnectedness today, haven't we? From the exquisite, really delicate balance required for our T cells and cytokines to function correctly, through the powerful, almost catalytic influence of IL-6 in driving pathogenic TH17 differentiation and those dangerously amplifying positive feedback loops, all the way to the devastating potential of full-blown cytokine storms. We've considered how IgG4's unique role in immune pathogenesis, even as an inferred contributor in IgG4-related diseases, might set a crucial chronic pro-inflammatory stage, effectively priming the system.
Setting the stage is a good way to put it.
And crucially, we've hopefully understood how foundational immune escape mechanisms ultimately pave the way for these complex self-attacks to even begin. They provide the very cells that can eventually turn rogue. What stands out, I hope, as a powerful aha moment for you listening is not just the sheer complexity, but the exquisite precision required for our immune system to function correctly day in, day out, and how seemingly minor deviations or these cascading effects, like those driven by elevated IL-6 and the expansion of TH17 cells, how they can, especially in the context of that underlying immune escape, lead to severe and systemic consequences, particularly in the medium and long term.
It truly is a delicate dance.
It really is. And when the music stops or goes haywire, the consequences can be catastrophic. So considering this intricate web we've uncovered today, from the molecular switches like STAT3 and ROR, right up to the systemic impacts like organ dysfunction, what are the broader implications here for the future of personalized medicine? How might understanding these specific interconnected pathways from potential IgG4 involvement, establishing that chronic pro-inflammatory state, to the precise mechanisms of the IL-6 TH17 axis and those foundational elements of immune escape, how might that allow us to develop not just new, targeted treatments that actually interrupt these cycles?
Rather than just treating symptoms.
Exactly. But maybe even truly predictive diagnostics, the potential to identify individuals at risk of autoimmune dysregulation or dangerous cytokine storms, perhaps years before they even manifest clinically, allowing for proactive, maybe even preventative interventions.
That would be the ultimate goal, wouldn't it?
It would. It's a powerful and hopeful thought to leave you with the potential to anticipate and maybe even prevent these complex self-inflicted attacks before they take hold. Thank you so much for joining us on this deep dive into the fascinating and sometimes, yes, frightening world of immune dysregulation. We'll be back soon with another exploration of the knowledge that truly matters.